Good afternoon, sir.
Madam ji, good afternoon.
Good afternoon, sir.
A hearty welcome to today's session. So, all the participants, please mute yourself and listen carefully. Dr. E. Madhuri Madam Garu, Assistant Professor, Department of Microbiology, Government Degree College for Women, Begum College. Madam has huge experience in microbiology. So, we will listen carefully. Madam, please you start the session.
Yes. Good afternoon, dear participants. Hope I am audible.
Yes, madam, you're audible. Please proceed.
Okay. So today we are going to learn about bacterial nutrition. So what is bacterial nutrition? Nutrition is something, the way the bacteria is taking the food. Okay. So microorganisms require 10 elements because in larger quantities to construct different components required by their cells. Sir, I'm changing the slide, but it is not reflecting in this video, I think. నేను మళ్ళీ స్క్రీన్ షేర్ చేస్తాను సార్.
Okay, madam, please proceed.
Okay. So bacteria need different nutrients. Okay. And out of these nutrients, some they may need in larger quantities and some may be in minor quantities. Okay. So the nutrients which are required in major quantities are called as macronutrients, and the nutrients which are required in minor quantities are called as trace elements or minor nutrients. Right. So here, what about macronutrients? Macronutrients are required in larger quantities, and 95% or more of the cell dry weight is made of few major elements. If you take the weight of the cell, dry weight of the cell, you can see that the cell is mainly composed of carbon, oxygen, hydrogen, and nitrogen. Okay, these major 4 elements. After that comes your sulfur, phosphorus, potassium, calcium, magnesium, and iron. So all these elements together are called as macronutrients. And the first 6 are components of carbohydrates, lipids, proteins, and nucleic acids. You might have studied about all these components, like the structure of carbohydrates, proteins, etc. And you might have come across, even you have drawn the structures where you could find the presence of carbon, hydrogen, oxygen, nitrogen, and sometimes phosphorus and sulfur also. Okay, next. Coming to the trace elements or micronutrients. As the name suggests, these elements or these nutrients are required in smaller quantities. But you can't say as these are required in smaller quantities, they are not important. No, they are very much important for the cell, but the required quantity is less. So where they are required? They're required for enzyme action as cofactors, coenzymes, etc. And apart from this, they also require growth factors, not only macronutrients, not only micronutrients or trace elements, they also require growth factors. Growth factors are these amino acids, purines and pyrimidines, and vitamins. So here, these amino acids are required for protein synthesis. You know, the proteins are the building blocks of the cell. Any cell, proteins are very much required. So sometimes you may have to give these amino acids in the growth media for the growth of the cells. And Similarly, purines and pyrimidines are also required. Where these purines and pyrimidines are present, they are present in the nucleic acids. What are nucleic acids? DNA and RNA. Okay, so purines are double ring compounds and pyrimidines are single ring compounds. They are the components of nucleic acids. And vitamins, these are organic molecules that act as enzyme cofactors, etc. So all these growth factors are also required by the cells for their growth.
Excuse me, madam.
And
Please click on the slideshow.
I have clicked, sir, but it is not reflecting on in the video. There is some technical
Okay, madam.
problem.
Okay, madam. Please you proceed.
The growth of microbes depend on supply of suitable nutrients, pH, oxygen, and temperature. If you feel like you have got the bacteria, right? You gave these micronutrients, macronutrients, and growth factors. Everything you have provided, and you ask the bacteria to grow. Okay, can it grow? No. Apart from that, nutrients or apart from the nutrition, it also requires some essential conditions. Okay, what are that conditions? It may be pH requirement, it may be suitable oxygen concentration. Are three. First one is carbon source. Whenever you are prepare a media or a food for the bacteria, you have to think about three main sources. One is carbon source. Okay. Next is nitrogen source. Carbon source. Second is nitrogen source. And the third is source of energy. Okay. ATP is also required for the bacteria to grow. And smaller amounts of molecules such as phosphate for nucleic acids and ions for enzymatic activity. Already we have discussed this in trace elements. And so now we have understood what are required for the bacteria or the microorganism to grow. What are required, ma? It requires nutrition, which consists of macronutrients, micronutrients, and growth factors. Okay. And apart from that, we also discussed it requires suitable conditions. What suitable conditions? It can be temperature, pH, oxygen, anything. So all the conditions you are providing, all the nutrients you are providing, then definitely the bacteria or the microorganism can grow. Right. But you have to understand that based on the nutrition, the cells are of different types. The microorganisms are of different types. Okay. So what are the different types? One is autotroph. Auto means self. Trophy means nutrition. Means the cells which can prepare their food material on their own are called as autotrophs. Okay. Autotrophs means these are the bacteria or the microorganisms which can prepare their own food. Like plants, what they will do? They will perform photosynthesis and they'll prepare their own food. Similarly, bacteria can also be an autotroph which can prepare their own food. Okay. So they can utilize very simple inorganic compounds like carbon dioxide as carbon source and ammonium salts as nitrogen source. They are also called as lithotrophs, obtain energy either photosynthetically or by oxidation of inorganic compounds. So we are saying that these are autotrophs. They can prepare their own food. So how they are preparing their own food? They can do it by photosynthesis or by oxidation of inorganic compounds. They are called as chemolithotrophs. They are using chemicals for their energy. That is why chemolithotroph. This inorganic element is involved. That is why the word lithotroph means nutrition. Any word, whatever you want to understand, you have to break that word and then you have to take the meaning. Next. Heterotrophs. Hetero means these are not autotrophs. These are depending on others for their nutrition. Heterotrophs means they are depending on others for their nutrition because they are not able to synthesize their own food, and they require carbon sources, energy sources, nitrogen sources, everything they have to be provided with. The same substances is used as both the carbon source and energy source. All bacteria inhabit the human body fall into the heterotroph category. Okay, so when you understand, whenever we are getting disease, okay, doctor will say you got some infection. So infection means with what? It can be any bacteria, right? If there is some bacteria in our body, what that bacteria is doing there, it is depending on us. It is taking the food material from our body. It means that is a heterotroph. Next, culture media. So till now we have understood bacteria can grow. We have to give some food material. We have to provide some good conditions, right? Based on the type of nutrition, we have divided them as autotrophs, heterotrophs. Yes, and now we have to talk about culture media. What is culture media? Yes, after going home, you will have lunch, you will have dinner. Parents will provide you with the food, right? So for bacteria also we have to give some food. That is nothing but culture media. The original media used by Louis Pasteur were liquid like meat broth. Robert Koch used the solid media. So it is not like microbiology has developed in a single day. It took years. Okay, they have developed many methods how to grow the bacteria in the lab. So they have done many experiments. First they used liquid media and then they have used solid media. Okay, and they have taken the microorganisms from different sources like water, food, and other materials, and they have grown that microorganisms in the lab. And one more thing is they require different media. You can't say all the bacteria, 10 type of bacteria, you take the same media and you grow. No, they won't grow. They will have different requirements for the this nutrition. So they require different media. Next type of culture media, it can be basal media, enriched media, selective media, differential media, and then transport media. So here you have to remember just a concept. What is basal media? Basal media is, it is the basic media which can be given by to the microorganisms. Like some microorganisms, they don't need much nutrients. If you just give minimum amount of nutrients, they will grow. That is basal media. And enriched media means you have to provide special nutrients. You have to provide special nutrients. For example, if a bacteria is producing amylase enzyme, you have to provide starch in the media. Then only that bacteria will grow and it will produce this amylase. So you are enriching that media with the starch. And next is selective media. Selective media means using the media, you are selecting the microorganism, means you are designing the media in such a way that only one particular microbe can grow. That is, you are selecting that microbe using that media. And differential media or indicator media means it is the media which can differentiate between two microorganisms. For example, hemolysis, alpha hemolysis, beta hemolysis, and McConkey media. This is also a differential media where it can be different. It can differentiate between E. coli and Enterobacter species. And the last category is transport media. Here, transport media means it is the media which is used to add the sample and take it or carry to other area where lab is located. For example, now we have gone through this corona phase. Okay, if in case if you have elder patient where we are not able to take the elder patient, we are calling lab people to home where they are taking this sample and they are taking to lab and testing. So somewhat like this, transport media means something media which is used for transport of the culture. Okay, this is blood agar and environmental factors influencing growth. So what are the different factors that is affecting the growth of the bacteria. We have studied now. It requires particular oxygen concentration. It requires particular pH. It requires particular temperature. Okay, so now we are studying about oxygen. So based on oxygen presence or absence, we can divide the microorganisms as aerobes or anaerobes. If it is requiring oxygen, we have to call it as aerobe. If it is not requiring oxygen, if it doesn't like oxygen, we can call it as anaerobe. Okay. Now, in aerobe also, there can be two types, obligate and facultative. You have to remember that obligate, whenever the word comes obligate or obligation, it means compulsion or compulsory. Obligation means compulsion. Okay. So when we are saying it, the organism is obligate aerobe, it means that compulsory it requires oxygen. Okay. And facultative anaerobes means it is the opposite of obligate. It is not compulsory. Okay. Facultative means not compulsory, means it can grow in the presence or absence of oxygen. If you give oxygen, it will grow. If you do not give oxygen, then also it can grow. So such organisms can be called as facultative anaerobes. And then anaerobes, they do not require oxygen for their growth. And again, these anaerobes can be obligate as well as microaerophilic. Obligate means again compulsory. Means these cells, they do not like oxygen. If you give oxygen, they will die. Okay. And microaerophilic means very less amount of oxygen is required for these microorganisms. Example is Clostridium perfringens. Coming to the carbon dioxide, some organisms require carbon dioxide for their growth, and some organisms like pneumococci, gonococci, they require 5 to 10% carbon dioxide. Means we have to provide carbon dioxide in the media, and such organisms are called as capnophilic. Philic means loving. Anywhere, philic means loving. Temperature, if they are growing at normal room temperature, they are called as mesophiles. And if they are growing at low temperature, they are called as psychrophiles. And if they are growing at higher temperature, they are called as thermophiles. Again, philes, Philes, philes or philic again means it is loving. Mesophiles means normal temperature, the organisms which loves the normal temperature. Psychrophiles, the organisms which love the lower temperature. Thermophiles, the organisms which love higher temperature. And coming to the pH, most of the microorganisms or medically important bacteria, they grow at neutral pH or slightly alkaline pH, which falls in the range of 7.2 to 7.6. And some bacteria can grow at acidic and some at alkaline pH. Okay. So, sorry. So this was about the nutrition required by the bacteria and the conditions required by the bacteria. Hope you all have understood the requirements for the bacterial growth. Okay. So here we are stopping, and next topic is bacterial growth. Okay, this will start today. Tirupati sir, hello. Right. I'll just take the introduction as we are having a little time today. So, what is bacterial growth? Bacterial growth, when you say a child is growing, means the child is growing in size. But when you can say bacteria is growing, already the size is very less. So, when you want to say bacteria is growing, it should increase in not in size but in number. Okay. Next. So, bacteria will also divide. Bacteria will also divide. Here, this is the process of bacterial division. First, the cell elongates, the DNA replicates, then cell elongates, and then cell divides and becomes two. This is the general bacterial division mitosis. And coming to the generation time. What is generation time? Generation time is, Generation time is the time required for a cell to divide or for population to double. When the cell divides, it is called as the time required for the cell to become double is called as generation time. Average for bacteria is 1 to 3 hours. So in how much time one cell will become two? It is about 1 to 3 hours. And E. coli generation time is 20 minutes. Means if you take E. coli, in 20 minutes, one cell will become two. In another 20 minutes, the two cells will become four. Likewise. So 20 generations can be carried out in 7 hours. Generation means the division of the cells. And it will give around, in 7 hours, it can give 1 million cells. See the growth rate. It is very high. And number of cells. Here in this picture, you can see number of cells. First, there will be one cell. It will become two. It will divide and it will become two. And then two cells will become four. Then likewise, the number will increase. So number of generation. This is the first generation where one cell has become two. You can say this is the first generation where a single cell has divided into two cells. Okay. And this is the next will be second generation, third generation, fourth generation, fifth generation. See the number of cells in fifth generation. There are many cells in the fifth generation. And coming to the exponential value. Exponential value means number of cells. Here, how many cells are there? Two. Means 2 to the power 1. Here, how many cells are there? 4. When 4 you can get, when you write it as 2 to the power of 2, you can get 4 cells. Then 8 means 2 into 2 into 2 means 2 to the power of 3 will give you 8 cells. Similarly, 2 to the power of 4, 16, to the power of 5, 32 cells. Next, this is the growth curve. Growth curve will have 4 phases: lag phase, log phase or exponential phase, stationary phase and death phase. Okay, lag phase, log phase, stationary phase and death phase. Here you may get questions. Okay, so remember the phases. Right. Here the cell number, initially there won't be much increase in the cell number. Exponential phase, the cell number will drastically increase, and then in stationary phase, cell number will be constant, and then in death phase, the cell number will come down or decrease. Okay. And here living cells will be more in growth phase. Stationary phase also there will be more, but in death phase, living cells will gradually decrease and dead cells will increase. Next, standard growth curve. Just now whatever we have discussed, it is the same thing, lag phase. Here, as we have seen, the number is not increasing. What is the reason? Here, the cell will prepare itself for division. It is the preparation phase, or it is the phase where the cells are adjusting to the new medium. Okay, and the length of lag phase means how long the lag phase will be there. It will depend on what type of bacteria we are growing and what medium we are giving, and the phase of culture from which inoculation is taken, whether the bacteria is active or not active, or it is sleeping, right? Size or volume of inoculum means how many cells we have added. That also influence the lag phase length, and environmental factors also. Whatever temperature is required, if you give optimum temperature, then lag phase may be very short. But if the temperature is not optimum, then lag phase will be long. Similarly, log phase. It can also be called as logarithmic phase or exponential phase. Why the name logarithmic phase is the number of cells will increase drastically. Okay, so the name logarithmic phase, the number increase will be in geometric progression. During this period, bacteria will have high rate of metabolism and they are very much sensitive to antibiotics and radiation. Next is stationary phase. Here nutrients become limiting, or waste products become toxic. Means if you remember stationary phase, the number of cells will be constant. Means in other words, the death rate is equal to division rate. The number of cells is constant. In stationary phase, after time, a stage comes when rate of multiplication that becomes almost equal. The new cells formed and the cells dying is equal. It may be due to the nutrients, exhaustion of nutrients or accumulation of toxic products. And next phase is death phase. Here, the death of the cells will happen at faster rate due to exhaustion of nutrients, toxic accumulation, or autolysis. Autolysis means cells will break. This was about the growth curve and measuring growth. How you can grow, measure the growth. How many cells are there in the given sample? How to grow? How to measure their growth? Okay, it can be done by different methods like the dilution plating, turbidity analysis, or a direct count with hemocytometer or optical counter. Okay, so these are the different methods. Here, one method is serial dilution. You have to dilute the contents in a serial fashion from one tube to another, and then you have to perform the standard plate count. You have to inoculate the culture into the media, and then you have to count the colonies. And you have to get the values of CFU, colony forming units. What is the full form of CFU? Colony forming units. Next is turbidity. When the growth happens, there will be more turbidity. Here you can see this is the clear broth, and this is the turbid broth where you are not able to see what is at the back. Here you can see the text where the tube is clear, but here you are not able to see the text. Why? Because turbidity has increased. This turbidity can be measured using a calorimeter where they utilize the principle of Beer-Lambert's law. And electronic counting, there is a machine for detecting the cell count using electronic counters. Similarly, direct microscopic count, here you have to put the culture onto a slide, and then you have to put the slide under a microscope, and these lines will be there. You have to count the number of cells in each square, then you can get the count of number of cells in the given media. Here, like this. This is the grid where you can count the cells. Okay, and this we have discussed in the last presentation itself. pH, acidophiles, alkaliphiles. Based on the pH, you can classify the microbes as acidophiles, alkaliphiles, neutrophiles, etc. And this is the pH range where different what do you say, examples are given. For example, as acidic pH, lemon juice will be more acidic, pure water is neutral, and soaps will be in alkaline range. Okay, temperature, this also we have seen psychrophiles, mesophiles, and thermophiles, and hyperthermophiles more higher temperature. This is the graph. As the temperature is increasing, the growth, just to see the growth. Okay, psychrophiles, they are growing from 0 to 20 degrees temperature. Mesophiles, here 15 to 45 degrees centigrade. Thermophiles 40 to 45 to 70 degrees, and so on. Salt concentration, um, they can be called as halophiles if they require more salt for their growth. And halotolerant means they can, if they can tolerate higher salt concentration, they are called as halotolerant. What is the difference between these two terms? As I said, philes or philic means loving, means they love salt. They need, definitely they need salt for their growth. But halotolerant, it is not like that. If they do not get salt, they can grow. But if at all we give salt also, they can tolerate and they can grow in that condition. Xerophiles, they grow in very dry environments. Okay, so this is the graph showing the growth of halophiles. Okay, similarly, oxygen requirements, microaerophils, anaerobes, aerobic, we have seen. Here, this slide, you can see these are obligate aerobes. As they require the oxygen, they are coming up of the tube of the media to take the oxygen. And facultative anaerobes means they actually can grow in the presence of oxygen, but sometimes they can also grow in the absence of oxygen. So some cells at the base. In the third tube, obligate anaerobes. They do not like oxygen. Definitely all the cells will go down. And aerotolerant anaerobes, actually they are anaerobes. They love the anaerobic conditions, but now okay. So here the cells, they do not like oxygen. They are growing at the base, okay, but they are tolerant to oxygen, so they can grow all throughout the length of the tube. And microaerophils, they can grow at a particular length, just below the upper point of the media. They can grow because they require oxygen, but in less concentration. That is why they are called as microaerophiles. Okay. So this was about growth and nutrition of bacteria. Hope you understood, all the students. I think it will be helpful for you. And as suggested, I'll pass on the PPTs to Tirupati sir. He can share in the group. If you have any doubts, you can ask now.
Any doubt, my dear friends? Yes. Any doubt?
Okay, sir. We are in.
Don't have any doubts? Okay. Madam?
Yes, sir. Okay, sir. We'll end the session here.
Remaining topic will continue tomorrow, or we want another more session?
Yeah, today nutrition and growth is over. I'll take up next topic.
Reproduction and economic importance. Reproduction, bacterial reproduction and economic.
Here, bacterial reproduction and bacterial growth is the same.
Okay, okay, right. Then the economic importance will explain tomorrow, madam.
Yes, next class.
Okay. Economic importance. In that economic importance, harmful and useful effects of the bacteria, plants, animals, and human beings.
Okay.
Okay.
Sure.
Thank you. Thank you, madam. Thank you very much.
Okay.